The Titin gene encodes for the largest known protein in humans called Titin, also known as connectin, which plays vital roles with structure, development and regulation of both skeletal and cardiac muscle. Variants in Titin, particularly truncating variants, are implicated in cardiomyopathies, the dilated and sometimes hypertrophic cardiomyopathies. Given the size of the Titin gene, namely 363 exons coding for over 38,000 amino acids, makes it more susceptible than an average-sized gene to yield variants, especially when using exome sequencing. There is a high burden of truncating Titin variants reported in the apparently healthy general population, with up to 3% of healthy controls. The expanded use of clinical exome sequencing is identifying incidental truncating variants, and discerning clinical relevance is challenging. Although the current ACMG list of 59 actionable incidental genes does not include Titin, the appearance of truncating variants as an incidental finding leaves clinicians in a difficult situation on whether to evaluate these further. In this study, Drs. Connolly, Landstrom and co-authors use amino acid level, signal-to-noise ratio and percent spliced in analysis to compare truncating variants. 3 cohorts, a large referral base of clinical exome sequencing performed at Baylor Genetics, rare population variants defined as mean allele frequency less than 0.001 in NOMAD and prior published studies on Titin, and a subset of Titin truncating variant positive, unrelated proband children evaluated for cardiomyopathy. The yield of Titin truncating variants was similar in NOMAD and clinical exome sequencing groups, but markedly higher in those evaluated for cardiomyopathy. Pathological hotspots were identified within the A band and N-terminal I band, which closely correlated with regions of high percent spliced in. Incidental truncating variants and population truncating variants did not localize to these regions. The authors then reclassified variants using the ACMG criteria and incorporating the signal-to-noise ratio and high percent spliced in. Those reclassified as likely pathogenic or pathogenic were more likely to have evidence of cardiomyopathy on echocardiography than those reclassified as FUSs. The authors conclude incidentally identified truncating variants are relatively common in the general population, as well as in children undergoing exome sequencing. The use of amino acid level signal-to-noise analysis found in the clinical exome sequence cohort were similar to background variants in the general population, while variants found in cardiomyopathy cases had clearly different localization patterns. The use of amino acid signal-to-noise ratio may aid reclassification of variants with better confidence of those truly being pathogenic or likely pathogenic.